Okay, so I guess we can get started now.
Yeah.
Okay, so welcome everyone to the first ever Plant Postdoc seminar series. We are really happy to start this seminar series specifically targeted towards early career researchers from our Plant Postdocs organization. So basically, we are starting out with giving opportunities for postdocs in plant related fields to present their most recent work. Uh, for example, a preprint they have submitted recently, or any published work 3 months um, before the deadline of our webinar applications. So we are very happy to start this, and we have two excellent speakers for today. But before going into the speakers and introducing them, I just want to tell you what we envision for this seminar series. The first is we plan to have two talks every month, mostly on the last Tuesday of the month at 12 p.m. Eastern time. And we are, like I said, we are accepting preprints and articles published in the past 3 months from postdocs within 6 years from their PhD. So this is just to start out. We might expand it later, but right now we are keeping it for preprints and articles published within 3 months. And also, I just want to emphasize this very clearly. We don't have a selection committee. What we do is we accept all the applications, screen it, and then randomly select speakers. So we don't evaluate any of the applications. We randomly select people to give their to present their work. So if you are interested, just apply, and then depending on the number of people, we randomly select 6 or 8 speakers in each series, and then they get to present. But you can keep applying more again and again, so you get a chance to present. And the two other new features that we are planning to initiate, and we want to see how people respond to this, is providing an option for peer review for the preprints particularly, and also start a half-hour chat with the speaker session after the talks. So our speakers are ready to be there for half an hour after their talks, so you can informally chat with them and also ask questions about techniques and other things they've used in their talks. And also, I just want to give a shout out to my co-organizer, Alison Barnes. So we have been planning this, and then Arif and Sandra from the leadership team in Plant Postdocs have also helped us to get this moving. And also, I'd like to ask you to spread this widely so that we can all come together in promoting the work of fellow postdocs. So with that, so this series we have 6 amazing speakers, and 2 of the speakers will be talking today. But in March 30th, we have other 2 speakers, and on April 27th, we have 2 more speakers talking about their recent work. And in March, we'll also start signing up for the next series. That will be another 4 months of postdoc seminar series. So if you are interested, you can apply in March 2021. So with that, I would like to introduce our first speaker, Ajeet Chaudhary. Ajeet is originally from Ayodhya, a historic city in North India. He finished his MTech in Indian Institute of Technology, Kharagpur, and worked as a junior research fellow in India before moving to Technical University of Munich in Germany for his PhD in the lab of Professor K. Schneitz, working on cell wall signaling inward in tissue morphogenesis in Arabidopsis thaliana. Ajit is mainly interested in the mechanisms underlying plant defense and development during adverse environmental conditions, and he wants to start his own independent research group in the future focusing on plant developmental biology. So with that, I would let Ajit start his talk.
Okay, so let me share my screen.
And also, Ajit, I just wanted to remind you,
Yeah.
you have 20 minutes for your talk, and then 5 to 7 minutes for the Q&A. Once you are at 18 minutes, I'll start waving through on the video, and then we'll know you have 2 minutes to wrap up.
Sure.
Okay.
So do you guys see my slide?
Yes.
Okay, so thank you, Sunil, for kind introduction and providing me this opportunity to talk about my recent work. And thanks to the Plant Postdoc Seminar Organizing Committee for putting up this wonderful seminar series. So today I'm going to talk about cell wall damage impairs tissue morphogenesis in Arabidopsis thaliana mediated by the receptor kinase STRUBBELIG. But first, I would like to acknowledge the people who has contributed in this work. So the first person I would like to acknowledge is the Jin Gao, uh, who put a tremendous effort during the initial phase of this project. And later on, Sia provided some of QRT analysis data, and Barbara provided some physiological response assays data. And next, I would like to thanks to all my former colleagues for helping me throughout my PhD and throughout this project work. And last but not least, I would like to thank my former boss, Professor Kais Nice, for helping me throughout PhD and supporting with his all the wonderful suggestions. And without his help, the work wouldn't have been possible. So now moving on, let's start with work from the project. So most likely you all have been seeing this humanoid form of pumpkins, and mostly on the internet or maybe sometime during the Halloween time. And being a development biologist, beside the aesthetic point of view, this humanoid or the change altered pumpkin or the round shape melon give me a lesson. The whole developmental process or tissue morphogenesis could be adopted. And the same time, they are conserved, as in the nature, if you let them grow to their natural shape, all the time they end up making their characteristic shape or the sizes. So basically, to achieve a particular shape or the size of the plant organs, there are three forces in the action. The first in the mechanoperception, and second one is the physical pressures like turgor pressures that propels the cells from the inside to achieve the cell elongation. And the third one is the biochemistry of the cell wall. So typically, the plant has several different tissue organs, and they all have their own characteristic shape and the size. For instance, here in the rosette leaf of the Arabidopsis, they are typical of the rectangular shape with the very flat nature. And if we keep this rosette leaf under the microscope, we will see jigsaw puzzle shape for pavement cells. And these pavement cells, they have either lobe region or the neck region and in the neighboring cells. And these lobe and neck regions, they are interdigitated. To accommodate the cell elongation process, lobe pushes to the neck region. And that means the cell wall in this region has to be of different nature. So cell wall is more stiffer in the lobe region and the more softer in the neck region. And that suggests cell wall has to be a very continuous remodeling in a very localized and a controlled fashion. So for instance, to achieve tissue morphogenesis, cell wall required to be softening to achieve anisotropic growth. But same time, if there is a pathogen attack, cell wall need to be fortified by accumulating secondary metabolite or keeping up the additional layer in the cell wall. So that suggests cell wall undergoes continuous change, and there has to be a mechanism to sense requirement of these changes. And this mechanism is known as the cell wall integrity sensors. So cell wall integrity sensing mechanism has basically three components. In the extracellular matrix region is the cell wall. That is a kind of hub for all the signaling event. For instance, release of TAMP molecule upon the pathogen attack, or acts as a physical barrier or the source of metabolite. And once these requirement of changes happen, then these changes or the TAMP molecule, they are perceived by the cell surface receptors molecule. And this receptor molecule later on activate the downstream signaling pathway, that cellular response. And again, depending on the requirement, It could be either in the form of cell wall loosening enzyme, for example, expansion or hydrolysis, or for the cell wall fortifications, release of secondary metabolite, for instance, lignin or cellulose or reactive oxygen species. And this whole cell integrity pathway is a feedback loop mechanism. And once this whole feedback loop is broken, that lead to the pleiotropic growth or developmental defect in the plant, or plant defense pathway could be completely compromised. So STRUBBELIG is a receptor kinase in the Arabidopsis thaliana that known to involve in the various tissue morphogenesis in the above ground. So here you can see in the absence of STRUBBELIG protein, the flower, they fail to make proper ovule, that is female reproductive organ, and in the flora meristem, they fail to maintain the proper cell division defect, and in the below ground, so in the root, so they fail to maintain the very strict and streamlined root hair cell patterning. So STRUBBELIG is, as I said, is receptor-like kinase. It is localized at the plasma membrane as well as plasmadesmata, and apparently it controls tissue morphogenesis in the floral organ as well as root hair cell patterning. And this pleiotropic defect in the STRUBBELIG knockout mutant suggests if STRUBBELIG mutant has some cell wall defect. And to address that, former colleague in the lab, Sadwadepalli, he analyzed the cell wall component with the FTIR analysis. And FTIR stands for Fourier Transform Infrared Spectroscopy. It's a very qualitative and simple technique. Once you put your sample through the spectrometer, you get a very typical spectrum, and corresponding to the each wavelength, the peak intensity correspond to the particular compound. So if we superimpose a spectra of wild type to the STRUBBELIG mutant, we can clearly here see that there's a difference, obvious difference between wild type and the STRUBBELIG mutant, and that suggests indeed there is some biochemical defect in the STRUBBELIG mutant cell wall. And to corroborate this observation, further on, I expose STRUBBELIG mutant to further cell wall damage. And I found out upon isoxaban treatment, that is the cell wall damaging agent, in the epidermis of the root elongation zone, the cells, they bulge out way faster than the Columbia wild type. Here you can see in the Columbia wild type, cells are still intact and elongated the way they are supposed to be, but in the STRUBBELIG mutant, they try to bulge out, they make kind of bulb. And that is because of weakening of the cell wall. So taken together, it suggests indeed STRUBBELIG mutant has the defective cell wall. So now the next question we ask, if STRUBBELIG has the cell wall defect, could it be because STRUBBELIG is involved in cell wall integrity sensing? So thereby it cannot maintain the cell wall. And to address that question, we took already well established assays in the Arabidopsis research. So one way to address that by the genetic approach, by mutating the cell wall biosynthesis gene. So since cell wall is the major component of the cell wall, sorry, cellulose is the major component of cell wall. So by inhibiting cellulose biosynthesis, we activate, or the plant activate, downstream compensatory mechanism. And it could be achieved by either by genetically mutating the cellulose biosynthesis gene, or by using pharmacological agent like isoxaban and DCB or thoxdomin A, and they all act on the SESA cellulose biosynthesis machinery on the plasma membrane. And upon destabilizing this SESA complex, cell respond to the particular physiological, produce particular physiological responses. For instance, release in the reactive oxygen species, or transcriptional change in the defense-related genes, or ectopic lignification of callose, or the increase in the phytohormone like jasmonic acid and ethylene. And to test that, first we look for the accumulation of reactive oxygen species in the STRUBBELIG mutant background, upon isoxazoline treatment in a time-dependent manner. And we found in the wild-type plant, they start accumulating ROS just after the 30 minutes of isoxazoline treatment, and that was not happening in the STRUBBELIG background. And though in STRUBBELIG, we could see slight increase in the ROS level after 90 minutes, but even after 120 minutes, it could not reach to the level of wild type. And further on, we did more physiological assays like the lignin and the callose accumulation, and we looked in the transcriptional change of defense marker gene, and all the data taken together, they all suggest STRUBBELIG is involved in propagating cell wall stress. So now we have STRUBBELIG involved in the two distinct pathway, one in the tissue morphogenesis, the developmental pathway, and the same time propagating cell wall stress response. So the question is how these two distinct pathways are connected, and what is the link between, and what is the mechanism behind it. And to address that, so in the receptor mediated signaling pathway, there is a canonical belief, so the way receptor kinases work. And so once upon the perception of ligand, these receptors on the cell surface, they undergo a spatial change by making either hetero or homomer, or by changing the phosphorylation. And in the next step, they recruit cytoplasmic protein and thereby activate the downstream signaling pathway for the cellular response. And once the pathway is activated, then the receptor undergoes clathrin-mediated endocytosis to either for the degradation of the ligand-bound receptor or for the recycling of the receptor back to the plasma membrane. So we plan to test if STRUBBELIG is also undergoes this canonical way of receptor-mediated signaling. And previously, we found out STRUBBELIG undergoes constitutive clathrin-mediated endocytosis using three different clathrin-mediated markers, FM464 and SYP43, as well as clathrin light chain. We could show that internalized STRUBBELIG EGFP, they were co-localizing with the clathrin markers, and that all suggests STRUBBELIG undergoes clathrin-mediated marker. So the first thing we wanted to test, our very first hypothesis, if STRUBBELIG upon isoxazoline treatment undergoes conformational change at the plasma membrane. And to figure out, we used the fluorescence-based technique called fluorescence anisotropy, and I will not go into detail about the physics behind this fluorescence anisotropy, and let me simplify it by if we have plane polarized light, and once this plane polarized light passes through a different kind of molecule, then again it tends to depolarize. And depending on the mobility of this molecule or the size of molecule, if the protein or the molecule is really small in the size, then it will depolarize more. That means there is a low anisotropy. And if the protein complex or the molecule is heavy and the less slowly moving, then it tends to less depolarize plane polarized light. And by measuring this polarization, we can infer if there is any spatial change in the protein of interest. But once we tag this particular protein with the fluorophore, for example, EGFP, is also tend to goes under the FRET phenomena. And upon the FRET, that means if a protein molecule goes, if it is tagged with EGFP and undergoes homomer formation, then there is a decrease in anisotropy. So decrease in anisotropy could be because of more bigger complex formation, or could be simply because the FRET formation. So having this nice setup in the hand, I went on to test our experimental setup by using some real samples. So for that, I used translational fusion of TMO7 tagged with either with 1 time GFP or with the 3 time GFP. So And then this TMO is the target of monopterus. It is expressed in the cytoplasm as well as nucleus. So we measure anisotropy of this marker line, and we found if the TMO is tagged with the one-time GFP, it has fluorescence anisotropy close to 0.37. But if it is tagged with the three-time, anisotropy decrease greatly, and that suggests there is a homofret going on between after adding 3 times GFP. And if there is one time GFP, so these molecules are have higher anisotropy, that suggests they are spatially more restricted. And because in the literature, it is known that free EGFP molecule shows 0.33, which is way higher than this tag one times GFP anisotropy, and that simply tells us one times GFP, TMO7 tag with the one time GFP has more spatially restricted environment. And the same time, we use 3 different surface receptor kinases, STRUBBELIG, Thesis1, and Feronia, driven with their own promoter, and we found all 3 they have different fluorescent anisotropy. Again, in the same line, they have different spatial movement on the plasma membrane. So in principle, technique works. So then we went on to test our effect of isoxaben on STRUBBELIG spatial movement on plasma membrane, and we found after the isoxaben treatment there is an increase in anisotropy of STRUBBELIG EGFP, and that trend we could also observe in the thesis, but that change was not that big, and the Feronia does not react to the isoxaben at all. And it was in the similar line that is known, Feronia does not react to the isoxaben mediated cellular response. So one thing was done, like STRUBBELIG react to the isoxaben on the plasma membrane. So next thing was to see if STRUBBELIG after activation upon the isoxaben treatment also undergoes clathrin mediated endocytosis. So I measured the fluorescence intensity of EGFP STRUBBELIG tag EGFP at plasma membrane as well as in the cytoplasm, and I found out after the isoxaben treatment there was more cytoplasmic signal in for the STRUBBELIG EGFP. And the same time, when I used FM464 as an endocytosis marker, I found there was an increase cytoplasmic FM464 puncta co-localizing with STRUBBELIG EGFP. And taken together, they all suggest after the isoxaben treatment or 5-hour treatment, STRUBBELIG undergoes internalization in a class-independent way. So and then the next thing was, if STRUBBELIG get activated and it undergoes the internalization in the class-independent manner, that means there has to be decrease in overall protein quantity in the cells. And to quantify it, we use again translational fusion of STRUBBELIG with the EGFP driven with its own promoter and in a time-dependent fashion. And we found at the 5-hour of isoxaben treatment, so there is a significant decrease in fluorescence intensity, and that followed up to 8 hours. And this decrease in fluorescence intensity of STRUBBELIG could be corroborated with its transcriptional change. That we also observe is after the 6 hours, there is a decrease in the transcript of STRUBBELIG after isoxaben treatment. So whole message here is STRUBBELIG get activated upon the isoxaben treatment, and STRUBBELIG is down regulated. So what is the physiological response? So what is happening if STRUBBELIG is get down regulated? That means can isoxaben treatment lead to the STRUBBELIG phenocopy? And to test that hypothesis, then I observed root hair cell patterning after the isoxaben treatment in the Columbia wild type plant. So here in the wild type plant, we can see the very streamlined arrangement of the root hair cells, followed by in side by side 2 non-root hair cells, and that streamlined patterning is completely gone in the after the isoxaben treatment. And that is very similar to the STRUBBELIG mutant. And these phenotype we could not see if we use isoxazoline resistant mutant IXR2-1. And the similar phenocopy we observe in the floral organ phenotypes. So after isoxazoline treatment, there is a cell division defect in the plant, there, as well as there is sepal and petal organization defect, and we could also see defect in the ovule formation after isoxazoline treatment. So to further characterize at the molecular level, we look at the downstream target for the root hair patterning, and we use the GLABRA2. GLABRA2 is a transcriptional regulator, so it is expressed specifically in the non-root hair cells and regulate expression of the transcription required for the formation of hair cells. So after using this GLABRA gene as a non-cell cell identity marker fused with the GFP. So we express in Columbia wild type, and we observe after isoxazoline treatment, the patterning of the glabra curves is also completely dysregulated. And that observation we could see, or we could produce with several other drugs. And we could also observe in the genetic mutant for cellulose biosynthesis, the very similar kind of phenotypic defect like Strubbelig 9. So, and these phenotypic difference induced by the isoxazoline, they are not correlated with in the general cell wall damage by using several other microtubule destabilizing drug as well as the osmotic changing drug. We could not reproduce this Strubbelig phenotype. So they all suggest indeed, this phenotype is related specifically decreasing of Strubbelig from the plasma membrane upon the isoxazoline treatment. So in the next idea, if Strubbelig undergoes downregulation upon isoxazoline treatment, is it possible by ectopically providing extra Strubbelig can we counter the effect of isoxazoline? And that's what we see using the ectopic expressing Strubbelig, we could counteract the isoxazoline effect in the root hair cell patterning as well as in the floral organ development. So that brings me conclusion slide. So with all this data, it appears Strubbelig is involved in the development as well as cell wall stress signaling pathway, and there is a differential amount of Strubbelig is required for these two pathways. In one hand, for the development pathway, there is a higher amount of Strubbelig activity is required, but same time for the cell wall stress pathway, lesser Strubbelig activity is required. So during the stress condition, Strubbelig is downregulated to conserve the energy for the protection of the plant, and this lesser amount of Strubbelig is enough to activate the defense response. So in overall, that's how Strubbelig, I mean, that's what at the moment we feel Strubbelig is coordinating between the plant development as well as plant defense. And with that, I come to the thank you slide. So now I'm happy to take any questions.
Thank you, Ajeet. Really nice presentation.
Thank you.
We have a few questions here in the Q&A.
Yeah.
The first one is from Rajneesh.
Yeah.
He's asking, are the cellulose levels affected in the submutants?
Yeah, so hello Rajneesh. So it's nice questions, and actually we did in the very beginning of our study, we also thought there is a change in the cellulose level, but we could not observe any change in the cellulose content as well as any other, be the pectin or the hemicellulose content. So at this time point, we are sure there is not quantitatively decrease in any of cellular component, cell wall component. So it might be the spatial arrangement of these components in the cell wall creating the damage.
Okay. So the next question is, how sub regulates GL2 expression? How subcellulose and root hair development is linked? The question is from Jose
Yeah. I know. I mean, he's expert in that field. And if you won't mind, maybe I would like to discuss this point in detail, maybe later. But for now, I can say, I mean, in STRUBBELIG mutant itself, expression of glabra is already altered. And changing in the cellulose component by this experiment we already know, even in the PRC mutant, which are the cellulose CESA6 gene. And this CESA6 mutant, they show substantial decrease in the cellulose content. And they, I mean, in our hand at least, we could observe there is a slight but observable defect in the root hair cell patterning.
Okay, so yeah, so we can discuss more in the casual chat session later.
Yeah,
but
absolutely.
I think we have time for a couple more questions.
Yeah.
There is one question from Jitesh Vijayan. Do you think sub interacts with RBOH for Ross generation?
So
He also says nice work.
Yeah. So it's again, it's a very nice question. And actually, in the follow-up study, we did some co-IP mass spec data, and we are still analyzing those data. But I think we have found some candidate relating with the Ross generation. But I'm sure we haven't found this particular gene, ROBH or ROBD. We haven't found it.
Okay, so maybe last one question from Deepak Bhandari. Is there any link between sub-monitoring cell wall integrity and using that sensing to modulate immunity?
Yes, actually, we tried to do several pathoassays, and we could not find any susceptibility in the STRUBBELIG mutant. So at the moment, we are not sure if STRUBBELIG are related with the plant immunity pathway as such.
Okay, cool. Thank you. So Ajeet, if you can go to the Q&A box and then see if there are any unanswered questions, you can just type the answers there. And also, I just want to mention that Rajneesh Singhal and Deepak Bhandari, these two postdocs from Michigan State University, they are actually reviewing your preprint, and they will be writing a report on that to submit for the
Thank you, Deepak. Actually, I know Deepak from his time in the Cologne at Max Planck, so I never got opportunity to meet him personally, but I know, I mean, he's working and he has done a lot of work in the plant immunity.
Yeah. Okay.
Yeah. Thank you, Deepak. Yeah.
Okay. So we can now move on to the next speaker. Ajeet, can you stop sharing your screen?
Yeah.
Jaime, go ahead and put your slides on. Okay, so our next speaker is Jaime Simbaqueba. Jaime is a plant scientist from Colombia with a bachelor's and master's degree from Colombia. Then he moved to Australia for his PhD at the Australian National University in Canberra, working with Fusarium, oxysporum, tomato, Cape gooseberry pathosystem, supervised by Professor David Jones. After his PhD, Jaime moved back to Colombia in 2018 as a postdoctoral scientist and research staff, working with Fusarium oxysporum in different hosts at the Colombian Cooperation for Agriculture Research, AgroSavia. So yeah, you can take on, Jaime. You're muted.
Sorry, I had to stop sharing, but again, I will share. So thanks very much, Anil, for that introduction. We'll start with my talk. I will talk about some novel effectors that we found in work in Physalis peruviana, or Cape gooseberry, that well, we did during my position back in Rosario. So let's just start. Cape gooseberry is a solanaceous plant and is very important for Colombia. And it is mainly found in the South American region of Colombia, Ecuador, and Peru. And This is known as a superfruit. You can find in the supermarkets as a dry product or even in a fresh presentations. Now is found in Europe, but also United States mainly. So let's continue. This crop plant has a very limiting problem, which is caused by the Fusarium oxysporum, known as forma specialis physali, which causes vascular wilt. And as I mentioned before, for Colombia, this fruit is the second economic product in terms of fruit. And as you can see here in this square, healthy plants during production stages. And then compare with this other panel, you can see some wilted plants, and then in a closer view, you can see the symptoms of this wilting coming across in the vascular system. Then back in 2010, it was isolated a highly virulent strain. We call it MAP5, and then we could recreate the symptoms at the glasshouse conditions. So just to talk about a little bit of Fusarium oxysporum, this is a species complex, and they are grouped into forma specialis. And just to show a little phylogenetic relationship here. So strains the group of strains that infect melon are different from the ones that infect tomato, and they are not like those cannot infect the other host, and vice versa. And the strains that infect gooseberry are a little bit far away, but they are just specific for each host, as the same as the banana. So from the Fusarium oxysporum lycopersicid pathosystem known as Foph, there is an extensive evidence of information, and we know that the core, the genome is divided in core and lineage specific, or nowadays called adaptive genome, because this has the genes involved with pathogenicity, and also those in host specificity. So those genes often encode for small secreted proteins that are known as 6 in the case of Fusarium oxysporum, and are mainly effector genes. So in tomato, there are also resistance cultivars that have been generated by the combination of immunity genes that are responsible for the recognition of some of these effectors. What about the pathosystem and Cape gooseberry? So in Nagrosavia, there is a large germplasm, which has around 74 accessions of plant. Some of them are resistant, and other ones are susceptible. And we set up an experiment back in the days with infecting susceptible plants and resistant plants with the highly virulent strain, and generate an RNA-seq analysis, and just focusing in the differential gene expression, comparing the resistant plant and the susceptible plant. So we took root and stem cDNA libraries and in 4 days post-inoculation. So I went back to I went to Australia to perform my PhD to study the mechanism by this fungus uses to infect Cape gooseberries. So I started with a comparison doing transcriptomics using the RNA-seq data, comparing the reference genome, which is the one from Foph. And I just want to show you here the first result from the chromosome 14, which has most of the effectors that have been identified so far in Foph, and compare with our RNA-seq data, and found that some of them are highly identical between these two forma specialis. And so, for example, this cluster of genes, this cluster of genes from 610 to 7, they are highly identical, whereas this gene here is duplicated in Foph. So all of this evidence we corroborated by PCR in the actual DNA of Foph. So what about the function of these effector genes? At the time, there was evidence of D61 in tomato or fall, but there was no evidence of this cluster of genes. So we wanted to knock out this cluster of genes in fall, but we couldn't find any mutant. However, we continue with the functional characterization of these 61A and B further on. So I just want to show a little bit of this work in the 61. So here we have a phylogenetic tree of the 61 proteins, which is a protein that is highly present in several different forms specialis that affects different hosts. For example, here we have brassicas, solanaceous, bananas, and cucurbits, all are different or are more related to each other. And so, for example, in solanaceous plants, we have the 61B from Foph and the 61A, and we can see here clearly that this one 61B is closer to Foph 61 than the other solanaceous 61. So, brief story here. 61 is involved with pathogenicity. There are many knockout strains involved, and the tomato I3 recognizes this protein and confer resistance to the Foph race 3. So we perform functional genetics using the transcript of the genes of 61A and B from Foph and complement a 61 knockout strain in Foph. And so in this experiment, we didn't find any restoration of the symptoms of these complementary strains, but when I inoculated a tomato plant with the I3 gene, I was able to identify that 61B was recognized by the I3, but not the 61A from Foph. So these results were published back in 2018, and then I just continue with the project in Cape Gooseberries when I came back to Colombia. So as I mentioned, complete the Foph work, and I obtained a collaborative grant with Agrosavia and Los Andes and University of Los Andes. And so focus on the investigation of the Foph genome sequence to corroborate the findings, set up the functional analysis properly of the 61A and B in Foph, and then also explore some effectoromics of our genes that could be an I3 homologous in Cape Gooseberries that could lead to the resistance in Foph. So investigating the Foph genomics, I want to show you here some phylogenetic analysis at the genome level. So just to mention before, we sequenced the genome of Foph, and found that it is 44.7 megabase pair. We assembled in 1.k contigs. From these contigs, we found 14,000 almost different transcripts, and we used the single copy genes then to perform this phylogenetic analysis and identified or corroborating that the actual Foph strain is highly related to some other solanaceous infecting form specialis of oxysporum. So then here in this figure, I just want to highlight that the fact that the actual genome assembly is highly fragmented. This version. However, we can see clear that this also has the actual spaces of the lineage regions that were previously identified in the reference genome of race 2. Resemble the division of core and lineage-specific regions, and here we can see some regions that are not shared with another form specialist that could be unique to Foph. So this work was recently published in Frontiers. So moving on, I also found some novel putative effectors by using this genome assembly. So what I did here was use a combination of Signal P software and effector P, and found a couple of thousand proteins that are secreted, possibly during infection, and some of them are effector proteins, or could be effector proteins. And from those, I found 7 novel genes that could be effectors, and the remaining were homologous with different other protein specialities. I used the RNA-seq implanter that I presented before, and found that 6 of them were expressed during infection, and then these are candidates for further characterization. So some results here out of those homologs and novel effectors. I did some T-Blast10 search and found that, well, corroborate that the actual homologs were present in other additional strain sequence. And so here we have 5 of the strain sequence, and show that the effectors are only present in the pathogenic strains of Foph. So almost 100% identical from their counterparts here, but also the fact that the novel effectors are also only present as well in these non-pathogenic strains. So that suggests that this could be involved in pathogenicity, or also possibly in host specificity. So I performed a manual inspection of the genome regions, and specifically focused on the chromosome 14 that has the gene cluster that I mentioned before of the 610, 12, and 7, and just inspect a little bit more in the Foph genome assembly what was happening there. And because this genome is highly fragmented, so I just put pieces along, and then found that two contigs have this cluster of genes. I was able to assemble, and I also found that the NIM transposon elements are also present in this region, and this could extend it to another gene, which is a transcription factor that could be involved in the expression of those effectors. So that is really exciting, because there is strong evidence of horizontal gene transfer between these two closely related form specialities, and this is a really nice really nice result. And moving on, I also test or suggest an independent acquisition of those effectors genes found in Foph. And to test this, I used or I performed a phylogenetic analysis using the elongation factor alpha gene, and sequenced by Sanger in 39 Foph isolates, and found that these resemble the polyphyletic origin of Fusarium oxysporum strains in the clade. And in this case, in Foph, we can show distinct two clades out of these strains, or some of them have the pathogenic strains that have been sequenced, and distinct here this Foph 36, which is the non-pathogenic one. So as compared here with the genomic resources of Foph, we have the MAP5, which is the highly virulent one, from the one that has been sequenced first, and the one that we used to do the RNA-seq experiment, and the remaining 4 novel sequences found that the actual genomes, in the case of the pathogenic ones, are highly conserved, or highly identical, compared with the number 36, which is the one that is not pathogenic, and also showing that the effectors are not present in this strain. So that means that these effectors could be involved in host specificity. So lastly, I just want to show the plant expression analysis out of this set of effector genes compared with some housekeeping genes. And so we can see here that the homologs are expressed at 4 days post-inoculation, but compared with the novel ones, for instance, the effector 2, the effector 4, and the effector 6 are highly expressed during infection. So that means that we have another proof that these are good candidates to continue with the characterization. Just to finalize here, I just mentioned that we identified genomic regions in the Foph genome that could be involved in pathogenicity. So good proof of that. Confirm our previous findings and identified 6 new candidates that are unique to Foph strains. We managed to publish those results, and in Agrosavia, my colleagues continue with future work focused on the characterization of effectors. Currently, they are working in the 61A and B. They are generating more. There is a work generating more complete assemblies of these Foph strains by using a different technology, such as the long-read one, and the effectoromics project, which is there to test if some candidate or resistant genes could interact with those effectors, and why not could be implemented for breeding strategies in the future for this crop plant. So just to acknowledge my previous group in Australia, the Professor David Jones here, which is now back from the COVID time, to the Professor Alejandro Caro, now he is at the Universidad Nacional in Colombia, and Dr. Carolina Gonzalez, which is the leader of the project of Cape Goodberry and Fusarium oxysporum. Just want to mention that I'm looking for a job now, and just drop a message in my email account or even my Twitter username. So that's pretty much that I wanted to show you. Thanks very much.
Yeah, nice talk, Jaime.
Thank you.
So I think we have time for a couple of questions. So you can go ahead and post questions in the Q&A. I have one question here in the chat by Miguel Acosta. He's asking why you're not working with mitochondrial DNA for the identification of Fusarium species.
That's a very good question. Well, mitochondrial DNA is highly conserved in all Fusarium species. Actually, Fusarium oxysporum species complex, so that we can just distinguish between strains. So that's why we use the genomic DNA. The genomic DNA.
Okay, so Jaime, I had a question for you. This is more general question. So I was thinking when you talked about core genes and lineage-specific genes, so you're talking about those in tomato and the Cape gooseberry, right? So what part of the genome, is it the genome that has the core genes or the ones which has lineage-specific genes, which are more important for resistance to Fusarium oxysporum?
Well, just to answer the question first, the lineage-specific genome or the adaptive genome and the core genome, I was talking about the fungus side or the pathogen side. Nothing to do with the plant or with the host side. So the lineage-specific region has all the effector genes, all the genes that are possibly involved in the virulence of the pathogen. Whereas the core genome, which is the most of the genome, is involved in other different structural things of the organism. So I don't know if that responds to your question.
Yeah, sure. Okay, sorry, I got confused, so that's why
Yeah,
I thought you're
that's fine.
talking about the host side.
Yeah, no, that's fine.
Okay, I think there's one more question from Jaime Pike.
Right.
Were any of the novel effectors you found not associated with MIMPs?
Yes, there are. I can't remember how many of those six novel effectors are associated with MIMPs. There's 5 of them, as far as I remember, but there is a table on the paper that we have that information there, where I identified MIMPs part of this novel effector. So they could be even the so-called 6 type genes or 6 type effectors. Yeah.
Okay, thank you. I think that's all the questions. I don't see any other questions. So I just wanted to let everyone know that in the chat, I have put the Zoom link for the meeting with the speakers. So that session will not be recorded. It will start in 2 minutes from now. And then we'll just be talking to Ajeet and Jaime about their work or in general about their postdoc experience and things like that. So if you are free, please join us there for an informal chat with Jaime and Ajeet. So with that, I would like to thank Alison for her help with organizing these webinars. And also I would like to mention that next month we are having another 2 good speakers for our postdoc seminar series. So please sign up again, and then you can listen to their talk as well. And then again in April, in the end of April, we have 2 more speakers talking. And then, like I mentioned before, in March, we'll open up again for applications for the seminar speakers. So with that, thank you all. I'll be on this Zoom for a couple of minutes, so everyone can transfer over to the other Zoom meeting link. So if you're moving to the other meeting, just wait for a minute for me to join there. And thank you, everyone.
Sunil, one more thing. If anyone in our participants group today is interested in helping us to moderate the next webinar so that we can hopefully get more people involved with our seminar series, you can send us an email to our Plant Postdocs email, letting us know that you would be interested in being a moderator, and we'll get back to you.
Yeah. Okay. Thank you, Alison. I forgot about it.
Yeah, that's okay.
Okay. So finally, thanks to the speakers, Jaime and Ajeet, for a wonderful talk today. And we'll meet you in the other Zoom. Okay. Bye.
Yeah. Bye-bye.